All right, now we have our time for our group discussion. We have until 10:30, so about 20 minutes. So happy to take questions for any of our panelists or speakers this morning. Amy, oh, okay. So did you have your hand up, Amy? No. So Mary and Eric? Okay.
I guess this is for Miriam. So in your data where you found methylation was a good predictor of blood pressure, that was adjusting for smoking and age?
Yes, yes. Yeah, we try to, because of the confounding I talked about, I think we have to adjust for quite a bit, which we wouldn't, for example, in GWAS.
Right.
So my question's basically for all of you, and it's quite handy and cute. You're all sitting together there. It's a good job. Measuring the environment is just notoriously difficult in free-living humans. And to compound that, we often don't know what the relevant environment is. But these multi-omic measures, whatever they are for each of you, in some ways they capture both genes, environment, and the interaction, multiplicative or other, unknown. And then, so really what we care about is how they predict disease. And I'm not sure if my question's biological or statistical. Is just, we as a field in NHGR High as an institute has struggled with how to do gene-environment interaction. And is there any way we can use these multi-omic measures and risk scores to get a hint about the importance of gene-environment interaction as it relates to human disease?
Well, so I'm not positive we get at gene-environment interaction, but I think it is definitely possible to use these measurements and a lot of existing biomarker measurements to develop proxies for environments that we don't know and maybe can't otherwise measure. And so when we talk about genetic prediction, the white space around the genetic prediction involves error measurement, but it also does involve environmental exposures, and it involves also random, truly stochastic factors that have influence on things that we measure. Some kinds of measurements, and I'm thinking a lot of the immunome space, you can't predict these random stochastic things, but you do see the consequences of stochastic variability, for example, in the development of the immune system, by what we can measure in T-cell repertoires and immunoglobulin repertoires. So you see the consequence of some of those stochastic factors in building the immune system. And I really do believe that we need to do more with the residual of our genetic measurements to build things that are capturing environment and some of the consequences of other kinds of stochastic factors that we maybe don't have measures for. And I think the metabolome is a great example of a combination of things that have high heritability, but some things that have little or no heritability. Some of those may be about exposure primarily. And so trying to use combinations of these omics and residuals of these omics and biomarker measures. I mean, think of the residual on liver fat. I mean, that's telling you some really important things about diet and excess calories. The residual on BMI, the residual on lipid levels, but some of these other measurements. I think there's a lot that can be done in that space, especially when we have data sets with those measured and some environmental measures that we can at least try to proxy with some of these things.
Can I give one, I think, very illustrative or important kind of anecdote? So my colleague Clary Klish was working with Bruce Spiegelman trying to identify endogenous ligands for various nuclear hormone receptors. I forgot if it was PGC1 or the PPARs, right? I mean, these are master energy regulators. And they thought in this fraction, they thought they knew what they had, and then they realized when they did a control that it was actually just the test tube alone. Something in the test tube was activating this. And it turned out that, it was a complete, it was a plasticizer. I think if that's not a motivating story to tell you, boy, we should be measuring the exposome in a real systematic way, as best we can. I think it's super important to do. So it sounds like a trivial example, but I think that's like a very important publishable paper.
Well, I think the example I gave with smoking, I think is an obvious one where you can have a set of, let's say, I don't remember how many there were in that prediction, but I think maybe 1,000 CPG, information on 1,000 CPG, and have an excellent proxy of smoking. In this case, it was current smoking, but there are other studies that show that even with that discriminator of current versus non-smoking, for example, it has also information on former smokers. And so you can really see that with 1,000 CPG, you capture information on smoking. So obviously smoking is an easy variable to get in studies. People report it fairly accurately, but there may be others that could be developed in the same ways, and that can really be a good proxy.
That would actually be a very interesting kind of model to test across all of these. So we measure cotinine in positive ion mode mass spec. You can look at various levels. We've done GWASs in small numbers, lots of interesting just subgenome-wide things in the brain, genes in the brain. You could integrate it with those, all the expression data. I mean, that would be like a proof of principle to examine gene and environmental interactions. We also see CYP genes and things like that metabolize. And there have been GWASs for cotinine as well. That would be kind of a nice proof of principle.
Peter?
So one of the opportunities I think we have right now is the ability to use data on hypothesized intermediates to summarize high-dimensional genetics or exposures. So I'm going to stick with the sort of transcript, the TWAS idea.
Closer to the microphone?
Yeah, sorry. So just sticking with genetics, but you could think of this as high-dimensional exposures as well. But if you have an ancillary study where this particular set of variants near such and such a gene are influencing the expression of that gene, and it's the expression of that gene that's really what's important, you can build that information into your prediction modeling. So actually, Mark alluded to this a little bit with a sort of fused lasso, you can put sort of priors on what you think the genetic architecture should look like, which you've learned from the mechanistic studies. So that's not something you can do. Like, it might be that it's gene expression in the brain that's important, right? We can't do that for prediction modeling. But if we have the subset where we've trained those models, we can use that information to help improve our prediction models. And there's some evidence, Alcus Price's group down the hall from me have shown that you can start to boost predictions by leveraging what we think we know about the underlying biology. Nilanjan Chatterjee has some papers on that as well. So I think that's sort of the opportunity. It's, again, on some level it's getting at underlying mechanism, but on some level we're still sort of just learning from the data.
Naomi, did you have a question?
Well, first of all, I've got a comment. So partly answering your question, we have a paper on bioRxiv where we've tried to evaluate G by E in the context of UK Biobank quantitative traits. And so this is actually Zhan Yang's work. So this is using variants as the trait. So can you find QTLs, which explains variants of the trait? And so that's quite interesting. And finding the evidence for GBIE for BMI, for example, but not that widespread actually. But I had a question for the panel. So for our genome-wide association studies, part of the success is having the very large sample sizes, and we've got historically lots of blood samples sitting around. But those blood samples have been collected in people, at many stages of their disease. Is it worth doing metabolites, lipids, DNA methylation on those samples or not?
So for DNA methylation, I think the temporality has to be carefully assessed. If you're looking at DNA methylation after you have had disease for some time, I'm not sure what you're going to be getting out of it. I would think that for DNA methylation, having longitudinal measures of DNA methylation would be fabulous. I mean, that's something that I dream of having, because we could, start to do something really interesting versus having this confounding issue that I've been talking about. But certainly temporality for DNA methylation is key. I think has to be very, very carefully assessed.
Well, no one else is answering. So if it's key for DNA methylation, does that mean it's key for metabolites and lipids and just not worth doing?
I certainly think it's going to be kind of context question dependent. And so for example, a lot of those samples were not obtained so first there's the biological question that you're after. The second is how the samples were handled. In general, though, for many metabolites and proteins, they're strikingly robust. So amino acids are incredibly stable. We've done a lot of studies of leaving them out on the countertop and then freezing them down and doing analyses before and after that. So I think that they are. So many compounds are very stable. You're going to lose. It's going to bias you towards the null, but many compounds and proteins will be stable under pretty reasonably extreme conditions.
So, I think that's true. I think I'm going to underscore Robert's point. So many of these are very stable in terms of sample handling. Some aren't, like carbohydrates. If you ship them overnight, that's it. They're gone. But I think the temporality is also a concern with metabolites, and it gets tricky pretty quick. So we did a study of the metabolite profiles of pancreatic cancer, and we saw branched-chain amino acids were associated with pancreatic cancer risk. But the story, after doing some biological mouse models that we came up with, is that it was actually an early stage marker of disease. So that we were picking up muscle wasting that was happening before they were diagnosed with pancreatic cancer. So they were latent cases. And you could see that, like the more distal the blood samples were from cancer incidence, the weaker the association, because it was further out and you weren't picking up this latent disease state. So yeah, I think it's challenging. I mean, the flip side is, if you were looking for something that could serve as a early biomarker or a diagnostic, or in the case of venous thromboembolism, when you come in and you're having a VTE, stuff goes crazy in your blood, but that might be useful for clinical outcome, right? So it depends on what the question is.
I have to say, we're very influenced by the genetics, so we're really trying to do the triangulation, MR experiments for every analyte that we measure to get at pathways.
How sensitive are those analytes to time of day? In other words, fasting, non-fasting, morning, evening, all that stuff?
Depends. I mean, but in general, most of the large cohort studies were done, the samples were done in the morning in fasting conditions. It really depends upon the analyte, and it's going to bias you towards the null. If you're looking at potentially a Mendelian disorder in children where you're going to see huge difference, then it might be informative. Obviously, that's going to bias you towards the null. A number of people have done some studies looking at that. Maybe Peter at the School of Public Health.
Yes, I don't remember off the top of my head. I'm still going to go with it. It varies from marker to marker, but I'd be happy to share our pilot studies on that.
Terry.
So, Miriam, I was surprised, and probably you were too, to see the very high AUC, probably the highest one I've ever seen, for smoking and for anything, but for smoking and this methylation measure. It sounds like you're looking for other things that are nearly that good, but do you have an idea as to why smoking is so strong with methylation?
I think, so looking at the EWAS studies we've done, there is the how can I say that? There is a persisting signature of smoking on the genome. So in other words, even if you stop smoking for a long time, you still see some epigenetic signature that are persistent. And I think, especially if you look at current smoking, that degree of association, that proximity of that association, perhaps is what is captured in these AUC curves. So, you have a very proximal measure of current smoking because essentially that exposure, even if you, let's say, for example, if you haven't, so you're a current smoker, that epigenetic signature is going to be fairly strong. Even if you don't smoke for a couple of days, that's probably going to be still the same. And that may not be the case, for example, for alcohol, where you may have this variation of day-to-day variation may not persist on the genome. And it seems like smoking, there is this persistent effect of smoking on the genome, and that's probably why you see this strong AUC curve. And that may or may not be the case for other exposures, but certainly for smoking, we see these very persistent signatures.
No, that makes sense. But you could think of other exposures that are invariant, like obesity. I mean, it does change somewhat, but day to day, no. And hormone levels or drug taking. I mean, it seems odd that one has such a big impact, and all the others are like 70 and below.
So we've seen in our EWAS of any traits, smoking had huge amount of signal. There is one specifically, and in fact, some have done some of this predictor of smoking with one CpG. That has a tremendous effect. And if you look even at the variance explained, I mean, in the smoking, it was 60% by epigenetic. So there is really truly that, some strong signature. And whether that's...
I'm not arguing that the signature is strong, it's why.
No, what I'm saying is that maybe, is that unique to smoking, or can we find others? Certainly, in all the EWAS we've done, certainly that one was... We started with that, but we got lucky because we had certainly the most signal from any trait we've looked at.
Yeah, but I mean, this is not a surprise. Because if you look at epidemiology, the biggest risk factor across a wide variety of disorders by orders of magnitude is smoking. And so in another, age, because we all die, that's fine, and we can do an G by E with age if you wish. But the reality is, if you wanted to find another signal, I think you'd have to do exactly what you're looking at. Look at the epidemiology and say, show me another exposure that across multiple diseases has that big of an attributable risk. If you did that, you would find exactly what you found in smoking, would be my prediction.
Well, I think what we're measuring also, you're measuring adducts that form methylation. Smoking causes adducts better than almost anything else. If you change the assay to something that's more relevant to obesity or relevant to something else, then you'll see that and probably have it persistent. I mean, you're using an assay to find what
you look
this
at
thing causes.
You look at obesity and metabolites, you see a tremendous signature, right? A similar, although we see pretty striking smoking signature on metabolism as well.
So another component is inflammation. And actually, we had a paper on Crohn's disease in Nature Communications earlier in the year showing that at diagnosis, you've got a massively strong methylation signature of inflammation. A year later, it's gone completely. And if you do the Mendelian randomization, you can see that the methylation at diagnosis is responsive to disease. And then that can be taken away. But then that's blood. But then there's also data saying that in the gut biopsies, that doesn't happen, that the signature persists. So I think it has to be longitudinal and tissue specific. I actually had a question for you. So how do you see implementation of, say, methylation age clinically for prediction?
Clinically, I'm not sure. Even for research, I have to say, it's a tricky prediction because. So yeah, I mean, I know Steve Horvath is very much pushing towards using this demethylation age for interventions, for anti-aging interventions, so designing this type of intervention. I'm not sure really what that means.
For plastic surgery, maybe?
I really don't know.
Wellness centers offer it now. It's quackery, but
right.
It is offered.
So how about including the methylation age in risk prediction equations instead of the chronological age, and then you could just check what predicts better. We do that all the time for measures of vascular aging, so why not just do the epigenetic age and then see if it's a better predictor? That should be, I mean, you have cohorts available where you could do that, right, Miriam?
Well, yeah, but I think, again, to me, that what the value of this is looking at the extreme, people who deviate from their chronological age and see what's specific to these people, whether what is it with them, what perhaps doing a PHEWAS on those.
That's a secondary question, but for risk prediction, you don't have to worry about that. You could just check whether
I'm not sure that you gain that much if you know somebody's age, chronological age.
But
I mean, those predictors do very well in predicting chronological age. So presumably, if you know chronological age, why would you use? And you're doing a prediction of whatever, a disease or whatever. Why wouldn't you use that? And what
you're saying instead of right now, chronologic age is a weak factor in a lot of equations. So whether it's calculating serum creatinine, predicting warfarin dose, whatever it is, age is in the factor. When we did the analyses that found out that VCOR C1 was replaced race as the predictor of warfarin, race fell out of the model completely, and the real cause, in this case VCOR C1 mutation, went into the model. Age is still in that model. Age should be taken out, but we just don't know what to replace it with. Maybe it's this.
Right, or age is in the model. This is some sort of summary of lifetime genetic and exposure history. Don't know what it's capturing exactly, but it works. Like, if we're just thinking like prediction and biomarkers, you could put them both in the model and see what happens. But I guess I wonder if part of your hesitancy has something to do with actual implementation. Like, are there challenges in measuring it? Is it touchy? Do you think it's just not ready to roll out?
Well, we don't really know, as you said, we don't really know what it captures. So
we don't know what the polygenic risk score is capturing.
No, I think we know a little bit more than what it captures.
Then great. If you're thinking prediction, it should be on the table.
It would be, why not look? We're scientists, right? So why not look? And particularly when you have somebody whose methylation age is so different from,
right. So,
I mean, those are the people who are interesting,
yes,
right?
Yes.
Right. I'd like to think the people who have younger age too would be interesting, but yeah.
I think those people are interesting. I think that would be worth looking at these people. What can we find out about these people?
Although the confounding by smoking is going to be a problem, because smoking is a premature ager in almost every tissue, right?
Right.
Yeah. So are you just measuring smoking? You'd have to do it in non-smokers, I think, to be sure, or at least stratify.
But for prediction, that might be good. Yeah. You could be capturing stuff that self-report wasn't getting.
One of the interesting things with the methylation is that you're capturing both. In some sense, some of this is affecting gene expression. That is, that you would get the same kinds of predictions as you would from gene expression. That is to say, some of our EQTLs are actually acting because they affect methylation status at sites that are variably methylated in the presence of certain kinds of exposures. But you're also capturing the consequences of exposures. So you're going in both directions and in sort of accumulating information. We've been trying to look at the smoking signature at the consequences of the methylation status at the sites involved in the smoking signature, at what that does to gene expression from generic studies of methylation, and then using that information to find people in BioView whose constitutive natural expression is most like that of the smoking signature, to see this long-term persistence of the methylation signature, which you can see in the placentas of mothers who smoke, and to some extent in the cord blood of infants born to those mothers, what the phenotypic consequences of the changes in gene expression that might be caused by these methylation changes, what that does to the medical phenome. And the preliminary stuff is inflammation, longer-term inflammation, some increased rates of cancer that you might expect and might be a consequence of the inflammation even. But in the brain, it's all about reward system biology. Part of what smoking does is to reinforce reward systems, I think, and reinforce the addiction, because that's what for the brain, that is the signature. It's all about reward system, so alcoholism, substance abuse, those kinds of phenotypes.
Well, my reward system is saying that I need caffeine, and I just am noticing that it's 10:30 exactly.
So before you break
Okay.
us, we have a half-hour break because we anticipated people might need to check out. Would you rather come back in 20 minutes and move forward a bit, those who are anxious for planes and stuff like that? So can we go with 20 minutes and be back at 10 of 11?
10 to 11.
10:50, yes.
10
10:50,
10:50.
great. All right, see you then.
Thanks.